Dear students, in this video we are going to talk about monohybrid cross. Monohybrid cross was the first hybridization experiments carried out by Mendel. Mendel was a great scientist of 19th century and is known as the father of genetics. So this experiment was the first experiment of hybridization which was carried out on pea plant, that is the Pisum sativa. Before this experiment, Mendel first of all established pure line. Pure line means pure plants obtained from true breeding. First of all, Mendel obtained pure plants of a particular characters by true breeding. Pure line means the plants which produce same characters, which express same characters in each and every successive generation. Take an example. If there is a pure line considering dwarfness or tallness, that is a character of height in each and every generation, there will be production of pure tall plants if tall plants are being bred. If dwarf plants are being cultivated, then each and every successive generation will essentially produce dwarf plants. So from dwarf we obtained dwarf plant, and from tall we obtained tall plant in each and every successive generation. Then this type of product will be called as pure line. So Mendel first established or first produced pure line, and pure line has been preserved for further experiment. What he did, he produced pure line, and from that pure line plants, seeds were collected and preserved for his experimental process. Now, first of all, the first experiment of hybridization was named as monohybrid cross. In monohybrid cross, he wanted to observe the expression of one character, that is the expression of one gene. And you can see here in this figure, pure line was generated by true breeding. Pure line plant, that is tall and tall plants were crossed pollinated artificially, and products were all talls. Now this is the pure line. After getting established pure line, he carried out the first hybridization experiment considering the effect of one gene, that is the effect of or expression of one character. What he did, he took homozygous tall pure plant and a homozygous dwarf pure plant, and it were cross-pollinated. These two types of plants, these two types of characters of same plants were cross-pollinated, and progeny of or offspring of these plants were all tall in F1 generation. Now this is the hybrid plant. This is the heterozygous plants. So in the first experiment of monohybrid cross, he obtained all tall plants but that were heterozygous tall in F1 generation. Thereafter, these tall plants were selfed. After selfing, he found that there are some plants are homozygous tall and some are heterozygous tall and some are homozygous, that is the pure dwarf plants, in the ratio of 3 is to 1, that is the phenotypic ratio, and 1 is to 2 is to 1, this is the genotypic ratio. Now, what happened and what are the conclusion of this monohybrid cross in Pisum sativum which was carried out by Mendel? He concluded the result and he analyzed and interpreted scientifically, and after interpretation of the experimental results obtained from him by him, he formulated some laws, and that laws were known as the law of dominance and law of segregation. You can see here tall and dwarf plant when cross-pollinated, then the results were tall. And after selfing, you can see in this figure, there are some plants tall and some are dwarf. So, you know from the definition of gene and allele, one, you can remind your concept, you can retain your understanding from the previous lecture or previous knowledge in which we had discussed the concept of genes and alleles. What are genes? Genes are the nucleotide sequences. Genes are nucleotide sequences or polynucleotides which are responsible to carry at least one character from one generation to another generation. So, genes are unit of heredity. That genes are responsible to transmit or transfer characters from one generation to another generation. And what are alleles? Alleles are the two alternative form of a same gene. That two alternative form of same gene you can see here in the case of homozygous tall plant. Capital T and capital T are gene is a gene which contain two alleles, but these alleles get separated during the process of gametogenesis, and gametogenesis is the production of gametes. During the process of gametogenesis, that is the gamete formation, these two alleles of a gene get separated, T and T. So, a homozygous tall plant produce similar gamete, and a homozygous dwarf plant produce a similar gametes. After pollination, this will produce all plants which will be tall. So, one law is derived from this experiment of the dominant character. Here you can see tall is dominant character because tall is being expressed in this monohybrid cross, but dwarfness is being hidden. Dwarfness is being recessive. So in a gene, one allele act as a dominant character, and second allele is acting as a recessive character. Dominant character is being expressed in as observed by Mendel, but recessive character is not being expressed. So there is the concept of dominant character coming from this experimental results. Now, come on this experiment. Here, during the selfing, there is the segregation or separation of alleles, and this F1 generation plant when selfed, then during the process of gametogenesis, gamete is formed. One gamete is of capital T types and second gamete is of small t types. You know, there is gametogenesis in flowering plants. In sexual reproduction, there is gametogenesis, that is the pre-fertilization process, and the pre-fertilization process, what occurs? First, gametogenesis occur. There is a male gamete and female gamete. Male gamete produce male. A male gamete is produced in male reproductive organ, and female gametes are produced in female reproductive organ. Flower contain male reproductive organ as, for example, anther. Anther is a male reproductive organ of a flower where pollen grain is produced. Now, during the process of this gametogenesis, that is the production of gametes, these two alleles are separated in the form of capital T and a small t. Here also in the form of capital T and a small t. After selfing, these two gametes get fertilized to produce this pair capital T, a small t, capital T, a small t, and a small t. So, what we are seeing here, after analysis of this result, what Mendel found that there are some characters which was not expressed in F1 generation were re-expressed in F2 generation. So, law of segregation come from this experiment. That is the selfing of F1 generation. So, there are two sets of experiment, one for generation of F1 generation, that is the production of F1 generation, and second set of experiment was selfing of plants which were obtained from the first filial, that is the first generation. First experiments gives but one law, that is the law of dominant, and second experiment also provide us the second law, that is the law of segregation. Now, on the basis of analysis and scientific interpretation of Mendel experiments, Mendel himself formulate laws of what? Inheritance, laws of heredity. First law is known as law of dominance. This is the first law, and according to this law of dominance, Mendel states that traits are controlled by discrete units which are known as factors, and these factors occur in pairs. In a dissimilar pair of factors, one member of the factor is dominant and second member, that is the second allele, is recessive. Now, these factors we know these days as a gene. So, gene was introduced or gene was explained by Mendel in the form of characters, in the form of factors. These factors were responsible to determine the characters which were expressed in the first filial generation as well as in second filial generation. So this law of dominance also states that alleles can be dominant or recessive. The dominant alleles, if present, will always determine the characters. Tall is dominant over dwarf in pea plants. Now, second law. Second law is known as law of segregation or purity of gamete. According to this law, none of the alleles are lost. Alleles get separated or purified during the process of gametogenesis. The recessive phenotypes, that are the alleles, not appeared or expressed in F1 generation are reappeared or re-expressed in F2 generation without any loss of alleles. So this was the monohybrid cross which were conducted by Mendel. Mendel was a great scientist of 19th century. He conducted hybridization experiment on pea plants in the garden of monastery served as he where he had been served as monk. So these are the experimental results of Mendel, and on the basis of Mendel's experiments, that is the monohybrid cross on pea plants, two laws were derived from that this monohybrid cross, and that these two laws are law of dominant and law of segregation. Thanks for watching this video.